Hi guys and girls, how are you doing today? My name is Belinda and I will be your life science presenter for today's session. Hope you guys are having a nice day. I hope you're keeping safe and still keeping healthy. Okay, so let's start. Before we begin, please make sure your audio is turned out and your speaker volume is turned up as well. Please note that you're automatically muted when joining the session. This means that I will not be able to hear you. If you've got questions, please use the question box and write your questions in there. We'll answer them as we continue the session or at the end of the session. Okay, you can also download the presentation on the handout section, right? You are encouraged to ask questions during the session or leave comments, please. However, do not ask irrelevant or inappropriate questions. Please only keep the questions to the content that's being discussed today. If we did not get to your questions, please send an email to academics@impact.co.za and we will get back to you as soon as possible. Visit the Impact's website for recordings of this session at www.impact.co.za. COVID-19 online schooling past class videos. You will also have access to the videos of the previous sessions as well as today's session. Okay, so from now on, we will be focusing on revision. So I will be doing Term 1's revision in this session, some of the topics from Term 1, and then we will continue with the revision as the webinars go. Okay, so I will be starting with Term 1 revision, but only highlighting the important parts of Term 1. Okay, and then maybe from there you can go back and check your past paper that you've written in Term 1 and maybe go and retry it again. Okay, so we will be doing Term 1 revision. Okay, not all the topics. We will only be looking at the chemistry of life, cells, and mitosis. Okay, so we'll be only looking at the 3 topics from Term 1 today. Okay, so before we start, again, if you are struggling to hear me, please make sure your audio is turned on and your volume is up. Your speaker volume is turned up. Okay, if you're struggling to hear me, I'm going to give you a few minutes to sort that out. Okay, during the session, please do not be surprised if we switch the webcam off. We might do so that we can improve the connectivity issues. For example, if I'm breaking up or something, we might need to turn off my my webcam so that you can be able to hear me clearly. Right? So please do not be surprised if that happens. Okay? I'm just going to give you a moment to sort out any technical issues that you might be having, sound or something. I'm going to give you a couple of minutes to sort that out. In the meantime, please download today's slides on the handout section. Okay. And then I'm going to ask everyone to please put up their hands. If you can hear me, please put up your hands. Okay, I can see the hands are going up. Thank you. you may put them down. I'm going to allow a few more minutes again. Okay, one more minute. Okay, you can get a pencil and paper just in case we will need to write down something, or just in case I would want you to take note of something. Okay, the slides contain the summarized information from term 1. So sometimes I might ask you to take note of something. Okay, for your convenience, I have summarized everything for you. So you might need a pen and paper to take note of some of the things that I didn't include in the slides. Okay. Okay, so I'm just gonna give you a few seconds to get it ready, and then we're gonna start. Okay, before we start, I'm the only one who sees the questions in the question box. How did you find term 1's test? Okay, for those of you who wrote term 1 tests, how did you find them? Did you do well? Didn't you? How did you find them? Was it difficult? Which topic did you struggle the most with? And how was it? How was your experience? Because this is your first, some of you, this is your first year doing life sciences. So it might have been a bit of an adjustment for you. You were not familiar with the way questions were being asked, but in the question box, please just share with me your experience for the first test that you wrote. If you wrote it, how did you find it? Did you find it easy? Did you do well? How was it? Which topics did you find challenging? Can you please share your experiences with me on the question box? Okay. Okay, so from term 1, the topics that you did were the chemistry of life, where you looked at organic and inorganic compounds. Okay, and then you did cells, the building blocks of life. You did cell division, mitosis. You did plant tissue. Okay, and then from term 2, you started with animal tissue and you did a couple of systems. You did the skeletal system, you did the circulatory system, okay, and you did uptake of water by plants. So that was term 2. So term 1, these are the topics that you covered, okay? So today we'll be looking at the chemistry of life, cells, and their functions, and we'll be looking at cell division, mitosis. Okay, so okay, so I'm waiting as we are going. Can you guys please just share? I see some of you guys have sharing your experiences of the first test and what you found difficult, and if whether you did well or not. Please, you can leave the comments on the question box. Nobody will see them but me, so no one is going to judge you, okay? So just leave them on the question box for me. Okay. Thank you for those of you who are responding. Okay, so firstly, you looked at the chemistry of life. So you looked at a couple of compounds, okay, or molecules. So in term 1, you've learned about inorganic and organic molecules. So in order for us to grow and in order for our cells to grow and be maintained in our bodies, living organisms need inorganic and organic molecules. Okay, so in order for us to grow and maintain the cells in our bodies, living organisms need inorganic and organic molecules. So inorganic molecules are molecules that are made from natural geophysical processes. Okay, and then organic molecules are molecules that are made through chemical reactions. So these are made through physical processes or physical reactions, and these are made through chemical reactions. Okay, so this is one of the differences between inorganic and organic compounds. Okay, and examples of inorganic compounds that you did in term 1 were water and mineral salts. Okay, and then examples of organic compounds that you did in term 1 were carbohydrates, lipids, proteins, enzymes, nucleic acids, and vitamins. Okay, so you've learned about okay, I apologize for my webcam. It's so that you guys can be able to hear me clearly. Okay, so from these organic compounds, you have learned about the building blocks of each and every compound. Okay, and you've learned also the functions or the role that they play in our bodies or in the cells in our bodies, as well as some deficiencies that come as a result of a lack of some of these organic molecules. Okay, so we'll be quickly looking at that, and we will be looking at the type of questions that are asked with regards to this topic. Okay, let's first look at the types of organic compounds in the body. Okay, so we have carbohydrates, also known as carbs, in short, and we have lipids, we have proteins, we have nucleic acid, and we have high energy compounds, but this is not in your syllabus, so you don't need to remember this. It's not going to be examined. But carbohydrates, lipids, proteins, nucleic acids are very important. They will be examined. Okay, so let's start with carbohydrates. So carbohydrates include polysaccharides, okay, which are made from disaccharides. Okay, disaccharides made from monosaccharides. Okay, so the monomer or the simplest form of a carbohydrate is a monosaccharide. Okay, so just like cells are building blocks of life, and from science or natural sciences in grade 7 and 8 and 9, you have learned that atoms are the building blocks of matter. Okay, monosaccharides are the building blocks of carbohydrates. Okay, so monosaccharides form disaccharides, which form polysaccharides. Remember, mono means one, di means two, and poly means many. Okay, so monomers for carbohydrates are called monosaccharides. Okay, lipids consist of what we call triglycerides. Okay, and which are formed by glycerol and fatty acids. Okay, so the building blocks of lipids are glycerol plus fatty acids. So all these two combined together are the building blocks of lipids. Okay, and then we have proteins that consist of peptides as well as some enzymes. Okay, I didn't write it there. And the building blocks of proteins are what we call amino acids. If you can remember from term 1, building blocks of proteins are amino acids. Okay. Next, we have nucleic acids. We also went through nucleic acids in term 1, which consists of DNA, DNA being deoxyribonucleic acid, and we had RNA being ribonucleic acid. Okay, these are the two types of nucleic acids that you looked at in term 1, and the building blocks of both this RNA and DNA are nucleotides. Okay, Remember, a nucleotide consists of a sugar, a base, and a phosphate. If you can remember from term 1, nucleotides are made up of a sugar, a pentose sugar, a base, a nitrogenous base, and a phosphate. Okay, so lots or millions of nucleotides form what we call DNA. You will be learning more about DNA and RNA in grade 12. All right. For now, you just needed to know that it is the type, or it's the 2 types of nucleic acids. Right. Again, an important section in term 1 was biochemical food tests. Okay, so I will be looking at the type of questions that can be asked with regards to biochemical food tests. And you could have been asked these questions already in your test as well, but expect this. It's good to learn it, so because it might come in your final examination as well. Okay, so we're gonna first start by looking at testing for starch. Okay, so to test for starch, we need what we call an iodine solution. Okay, you can get this from various chemists or pharmacies, and with supervision, you can be able to conduct the experiment by yourself. All right. So in order for you to test if a particular food contains starch, I don't know if you've had it, when people are on diet, they want to avoid starch because apparently starch makes you big and so forth, and they don't like starch. So if you want to test if a particular food or food particle contains starch, you need what we call an iodine solution. Okay, we've used an example of a potato here. Okay, so we wanted to check if a potato contains starch. Okay, so we're going to use the iodine solution. If a food particle contains starch, it will turn blue to black. Okay, if a food particle contains starch, it will turn blue or black, bluish black if you put it inside an iodine solution. Okay, so food particles containing starch turn bluish and blackish if you put it inside an iodine solution. If it doesn't turn bluish or blackish, then it does not contain starch. Okay, so clearly by looking at this experiment of a potato, it does contain starch since it turned blue after we added an iodine solution to it. Obviously, this doesn't work with all food particles. Some particles we need to boil first so that we can break the cell membrane so that the iodine solution can be able to react. Okay, so with different particles, we have different methods of testing for starch, but for all of them, we use iodine solution. Okay, to test for reducing sugars, okay, which are also types of carbohydrates, we use what we call a Benedict's test. Okay, an example of a reducing sugar is glucose. Right, so if we want to test for reducing sugars, we use a Benedict's solution. Okay, put it inside a test tube. Okay, in this test tube, they've used about 2 cubic centimeters. 2 cubic centimeters. You heat it inside, or you put it inside heated water. And if the food particle or substance contained reducing sugars, it's gonna turn brick red. Okay, so in here they've put about 2 cubic centimeters of a substance containing reducing sugars, and then they've put about 2 cubic centimeters of the Benedict solution as well in there. Put it inside heated water, If the solution turns red brick, then it means it is a reducing sugar. So a reducing sugar turns brick red if it is added inside the Benedict's test. Okay, so this is how we test for reducing sugars. We use the Benedict's test. Okay, proteins. Okay, so to check if food particles contain proteins, we can use one of two substances. We can use the Burette reagent, or we can use Millen's reagent. Okay, a positive result. So this means if a food particle contains a protein, if you put it inside a Burette reagent, it will turn violet or purple in color. If it doesn't, it will turn blue in color. Okay, so this negative result means does not contain protein. If you put it inside a Burette reagent, if it contains protein, it will turn purple in color. If it does not contain protein, it will turn blue in color. If you use a Millen's reagent, if it contains protein, it will turn red brown. Okay, if it doesn't, it will turn white in color. So let's look at this. We were testing if whether this food particle or substance or whatever it is that we were looking at contains proteins. Okay, we can see that it's turning purple. It's turning purple when we add the Burette reagent into it. It's turning purple. If it's turning purple, it means the food particle contained proteins. Okay, you can use an emulsion test to test for lipids or fats. Okay, lipids are also known as fats and oils. To do that, what you can do is put the food sample with alcohol. Okay, mix it together and then take that mixture and pour it inside water. If an emulsion is formed, then the food sample contains fats, it contains lipids. All right, okay, if you've got questions with this regards, please send them to, in the question box. You guys, you are encouraged to send questions. Please, if you've got questions with regards to biochemical food tests or the organic compounds, please do type them in the question box, and we will look at them at the end. All right. Okay. Okay. You are also required to look at. I'm not gonna go through it now because you can go through this by yourselves. Recommended dietary allowances that you needed to look at, and you needed to look at vitamins and vitamin deficiencies. Okay, also you need to look at food packaging. Okay, the packaging or the labels that comes with food on what is found in the food particles, what organic compounds are found in the food, and vitamins are found in the food particles. Okay, now let's look at the example of questions that come, or typical questions that are asked from chemistry of life. Okay, let's look at this question. Study the table below showing results of different food tests. Okay, so this table contains results from food tests. Okay, this was food type X, this was food type Y, and this was food type Z. Okay, they used a Benedict's test and an iodine solution test on these food types. Remember, what is a Benedict's test used for? A Benedict test is used for reducing sugars. It's used to test for reducing sugars. And then what is an iodine solution test used for checking? Starch. Okay, you can use an iodine solution to check for proteins. It is used to test starch. So if an iodine solution is used, you know they were testing for starch. And if they're using a Benedict's test, you know they were testing for sugars. Okay, so just by reading the information that's given to you, if you have studied properly, you already have an idea of what has been done in this experiment. Okay, so they did the following. They used the Benedict's test on food type X, and it returned a blue color. Okay, they used the iodine solution test on another food type, but of the same type X, and they got a blue-black color. On Y, they used the Benedict's test, and the color that they got was orange. Okay, and then on the iodine solution test, they got blue and black. They tested particle Z again, and they got an orange color from the Benedict solution, and then they got a brown color from the iodine solution. Okay, the question comes, which one of the following types, X, Y, and Z, contains starch only? Okay, so looking at this table, okay, and I'm advising you guys to please do not be quick to answer questions. Always read them more than once. If you don't get it at the spot, read it 2-3 times. But before you answer a question, make sure you understood what it's asking you. Do not rush into answering a question. Okay? Because there's no point in you finishing quickly if you did not answer the questions correctly due to not being able to understand what it was asking you. So always take your time when you are reading the question. Make sure you understand the question before you answer them. All right. In your question box, please tell me which one of the following food types X, Y, and Z contains starch only. Let's look at this. This was blue. When put inside the Benedict's test, remember Benedict's test is for testing reducing sugars, and we know that if a food particle contains a reducing sugar, it turns blood orange. So this means this was a negative. It wasn't containing a reducing sugar. But looking at this iodine solution, we know that we were testing for starch, and because it gave us a blue-black color, we know that starch was present. So there was no reducing sugar, there was no glucose, but there was starch because it returned a positive result. Okay, so now they're asking you which one of the following food types X, Y, and Z contains sugar. In your question box, can you tell me if the substance that contains only starch, if it is X, Y, or Z? Can you type it in your question box? Can you please just attempt it? Okay. Which of the food types contains starch only? Okay, just attempt it. If it's wrong, oh well, you will know what the right answer is. Okay, but please just try. Okay, the food type that contains starch only is food type X. Okay, it does not contain glucose because it tested negative for glucose. Okay, Y contains starch, but it contains glucose as well. I'm probably giving you the answers to the other questions. This one does not contain starch since it turned brown, but it contains a reducing sugar. It contains glucose. Okay, so this means for question A, the correct answer is X. Okay, well done for you guys who got that right. Okay, how about B? How about B? Which of the food particles X, Y, and Z contains glucose only? Can you type it in your question box? I see some of you have already answered that question. Which one of the following food particles contains glucose only? Okay, thank you to all that answered. For question B, the food type that contains glucose only is Z. It does not contain starch because it tested negative for starch. Remember, if it does not have starch, a food particle will turn brown. If it contains starch, it'll turn blue and black. So Z is the correct answer for B. Okay, glucose and starch. Okay, Y, food particle Y contains both glucose because it tested positive for the Benedict test. It tested positive for reducing agent, and it tested positive for starch. Okay, if you've got questions, please type them on the question box. All right, this is a 4-mark question that was also asked based on chemistry of life. Farmers use fertilizers to promote plant growth. Explain the effects of excessive use of fertilizers. What effects do they have? But you don't need to answer this. You can please do this after our session on your own. Okay, explaining the effects of excessive use of fertilizers. Okay, for the sake of time, I'm not gonna go through that question. Okay, as well as this one. Let's look at this question quickly. We have a table that shows nutritional information on cereal packets. Sorry. On cereal packets, 3 cereal packets A, B, and C. Each packet has a mass of 500 grams. A 15-year-old boy showed the following symptoms after eating 1 serving of cereal B. He experienced bleeding gums, and nosebleed, and sores on the skin. Okay, I'm just gonna ask you question 3.2.1 only. What is the monomer for a protein? If you remember, guys, what is the building block of protein? Please type it on the in the question box. The monomer or building block of protein. Monomer and building block means the same thing. Okay, what is the monomer of protein, or what is the building block of protein? Please attempt it. Okay, somebody posted the correct answer. The building blocks of proteins are amino acids. Okay, let's go back to that slide that talked about this. The building blocks of proteins are amino acids. Building blocks of nucleic acids are nucleotides. Building blocks of lipids are fatty acids and glycerol. Building blocks of carbohydrates are monosaccharides. Okay. Thank you for that correct answer. This is another typical question that can be asked on chemistry of life, on enzymes. Remember when we did the scientific report? This is a scientific report. If you forgot how to write a scientific report, please go and access the video from the previous week and then attempt this question, attempt to answer this question as well. Okay, the aim, remember with a scientific report, you need to have an aim, you need to create or derive a hypothesis, state the independent and dependent variable, the planning steps, in an experiment, conclusion, and so forth. So please, if you forgot how to write a scientific report, go back to that video. You can find it on www.impact.co.za and just replay the recording again and then attempt to answer those questions. All right. Cells. Okay, so cells are the basic unit of life. So we just looked at the basic units of organic compounds, and we've seen that the building blocks of proteins are amino acids. The building blocks of life, building blocks of organisms are cells. So in order for us to be the beautiful girls and handsome boys that we are today, we started off as single cells, and they divided by mitosis, which you're gonna do next. They divided by mitosis to form tissue, continued to multiply until organs were formed, continued to multiply until continued to divide until systems were formed, continued to divide until a whole organism with a whole lot of systems was formed. Okay, so cells made all this possible. Cells are alive themselves. And they've got also, they have little organelles inside them that perform various functions to keep us alive. Remember, the inorganic and organic compounds that we ingest or that our bodies produce maintain these cells. They make it possible for our cells to grow. They make it possible for our cells to develop into tissues. Okay, and also if you are hurt or your cells die, they will continue to divide to repair themselves or to replace themselves if they are dead. Okay, and they do this for as long as you are alive. Okay, so who can tell me what type of cell is this? We have two types of cells. We've got animal cells and we've got plant cells. And by looking at the structures of the two, you can clearly see that there is structural differences. So this is a type of a cell, either animal or plant cell. Okay, if you can remember from term 1, is this a plant or an animal cell? And why do you say that? I see somebody said in the question box that this is an animal cell. Why do you say, or what do you see? What tells you that this is an animal cell and not a plant cell? Okay, it has no shape. It's got an irregular shape. Okay, this means it's got no cell wall. It's got an irregular shape. Okay, an animal, a plant cell has a regular shape and a cell wall. This one has an irregular shape. So that's an animal cell. Okay. So let's look at the structural differences and similarities between plant cell. Let's look at the differences first. There is the structure of a plant cell. It's got a much more regular shape because of that cell wall outside it. This is irregular. That's a shape of an animal cell. Please go look at the more detailed structure of a plant and animal cell in your book. Okay, a much more detailed one. Okay, so the differences between a plant and an animal cell is that with a plant cell, we have a cell wall. A plant cell has a cell wall surrounding it, which gives it a shape, which gives it a regular shape. An animal cell has no cell wall surrounding it, only a cell membrane. Okay. A plant cell has a large vacuole. Okay, it has a large vacuole inside it, whereas a An animal cell has a small or tiny vacuoles or no vacuole at all. Okay, this plant cell has a large vacuole. You can see it. You can see it just by looking at the cell. It is prominent. You won't miss the large vacuole. Okay, as we go, let's look at the functions as well. Okay, I'm going to ask you to write this down. The functions, or if you are able to, you can take screenshots. I'm going to write them here. Okay, functions of a cell wall, which is found in the plant cell only, is that it gives the plant its shape. It gives the plant cell its shape, not the plant. It gives, forgive my handwriting, gives the plant cell shape. Shape. Okay. And then we said in a plant cell, we've got a large, prominent vacuole. But what does a vacuole do? Okay, a vacuole acts as a storage system. It acts as a storage system for minerals and so forth. It acts as a storage system. Okay. Another thing that we have in plant cell that we don't have in an animal cell is a chloroplast. Chloroplast is only found in plant cells, and it contains a green pigment. Known as chlorophyll that is needed for photosynthesis. Okay. I don't normally write like this, you guys. It's cold and the technology is, yeah, it's a bit hectic. Needed for photosynthesis. Okay. This green pigment is known as chlorophyll. Okay, so chloroplast is needed for photosynthesis. Okay, we have flagella, but this is found in gametes only. Gametes, you will learn this in grade 12. These are sex cells. Sex cells, you will learn them in grade 12, so I'm not going to go into detail. This flagella is used for movement or for locomotion. Okay, so those are the differences between them. Okay, but both of them have mitochondria. Okay, you will learn more about this in grade 11 when you do cellular respiration. Okay, mitochondrion provides energy. Okay, this is for cellular respiration, where energy is produced. Okay, provides energy. Okay, and then we have a Golgi apparatus, which sorts materials. So it is a sorter, it sorts the materials. That's material. This is material. I'm so sorry. We have the ER or endoplasmic reticulum. This is a transport system inside the cell. This is the transport system inside the cell. It transports various things within the cell. Transport system. Okay, the vacuole is the storage system. Endoplasmic reticulum is the transport system inside the cell. Where did my pen go? Okay, nucleus is the CEO of the cell. It controls everything inside the cell. Okay, I can't find my pen. Oh, there we go. This is the transport system. The nucleus is the CEO of the cell, controls the control center. It's the boss. And then we have cytoplasm, which is a home for all these organelles. It acts as a secretary. It keeps everything together. Okay, it's a fluid-like substance, fluid that keeps or houses the organelles inside a cell. Organelles. Okay, those are the small little organs that are found inside the cell. Okay, ribosomes produce proteins. Okay, not the proteins that are found inside your food. Your body also produces proteins. Ne? So inside the cell, proteins are produced in the ribosomes. Okay, so you can take a screenshot of this. Otherwise, you can rewrite it if you think my handwriting is horrible. The problem is I'm going to be moving into the next slide now so that we don't run out of time before I explain everything. Okay. There's also lysosomes that are found in here. Okay. Lysosomes are recycle unneeded parts. Okay. Lysosomes are also found in here. Let me just write it there. They are recycling agents inside the cell. Okay, so a cell is alive and it's just like a factory. Things are happening in there. Just like in your house, there's a kitchen where food is cooked, and there's a sitting room where there's entertainment, there's TV, there's the bedroom where you sleep. Inside the cell, there is processes that are taking place that keep us alive. Okay, so from chemistry of life, you've learned that you need a couple of vitamins, nutrients, and organic compounds to maintain the cell. Yeah. Okay, as we said, ribosomes are the place where proteins are produced. So it's like a protein factory. Okay, the cell membrane is like a barrier line that controls what comes in and out. Okay, after proteins are produced inside your body, they need to be exported where they are needed. For example, in your muscles or whatever they are needed in your body. Okay, they will be exported. The transport system. Remember, what did we say? What we said the transport system is? It's the endoplasmic reticulum. It is responsible for transporting things in and out of the cell. Okay, the nucleus is the control center. As you can see, the boss of the cell. Okay, and this packaging plant or this blue cute little things that are packaging things. There will be our Golgi bodies or our Golgi apparatus. They sort things together, they arrange them together for safe transportation where they are needed. Okay, this is the energy machine where energy is produced. Okay, and we know that energy is produced by what? By the mitochondrion. Okay, so this is clearly a animal cell because we don't have a chloroplast. Otherwise, if it was there, that would be the place where sugar is produced, where sugar is produced from photosynthesis. But because this is an animal cell, we don't have the chloroplast in there. Okay. If you've got any questions, please do ask. Let me see if everyone is still here. Can you please raise your hands? If you can hear me, can I ask everyone to raise their hands? Okay, thank you. Okay, thank you. You may put them down. Okay, so that's basically what happens inside a cell. It's a living factory, tiny living factory. This is a typical question that can be asked in this topic. Okay, study the diagram of a cell below. This is a cell. It's got that organelle A, B, and C. Okay, now they're asking, is this a plant or an animal cell? You guys, can you type it in the question box quickly? Is this a plant or an animal cell? This is not a cell wall, by the way. It's just a border. It's just a border for my picture. It's not a cell wall. Okay, so this is the cell. This is the cell that I'm talking about. Is this a plant or an animal cell? Please type it in the question box. As I've said, this is not a cell wall, it's just a border for the picture. Okay, so because it contains no cell wall and it's got no shape, it's got an irregular shape, this is an animal cell. Okay, the question then asks, give 3 reasons for your answer in question 2.1.1. Now this question is asking, why do you think this is an animal cell and not a plant cell? Easy. Number 1, it's got an irregular shape. Number 2, it's got no cell wall. Number 3, it's got no chloroplast. Okay. Number 4, there is small vacuoles. Okay. If it was a plant cell, we would have a huge vacuole there. Let me draw it. Okay. Let me draw it. If this was a plant cell, first of all, it was going to have a cell wall and a cell membrane. It was going to have a huge vacuole. Okay, this one only has small vacuoles. So because it does not contain a large vacuole, it is definitely an animal cell and not a plant cell. And it's got an irregular shape and as well as no cell wall. Okay. Identify parts A and B. Okay, part A is the mitochondrion. Okay, part B is the nucleus. It's the CEO of the cell. You can't miss it. You cannot know the boss of this whole cell. Okay, state one function of part A. What happens in the mitochondrion? The mitochondrion is responsible for providing the cell with energy. Okay, it provides the cell with energy. Or this is where cellular respiration takes place. Well, explain what would happen to a cell if part C, the cell membranes becomes impermeable. Impermeable means nothing can come in. If this becomes impermeable, substances will not be able to pass through. So the nutrients that are needed by the cell to survive will not be able to pass through. This means the cell might die. With no nutrients, the cell might die. Okay, so this part needs to be semi-permeable, not impermeable. It needs to allow some substances to come in. If it becomes impermeable, some of the cells might die. All right. Okay, then we have mitosis. Okay, so in order for us to grow, cells need to divide and form more cells, and this process is known as mitosis. As I've explained in the first few slides, cells divide to form tissue, which divides to form organs, which divide to form systems, which divide to form the organism. And when you are ahead, a replacement needs to take place. Otherwise, you might get infections and you might die if cells that are damaged are not replaced, or if cells that are dead are not replaced. You might not survive that long. Okay, so mitosis is necessary for growth. Mitosis is necessary for growth. Okay, the cell division is necessary for growth. It is also necessary for repairing damaged cells and replacing dead cells. Okay, remember, mitosis is divided into phases, and we call this the PMET. PMET. Okay, interphase is not a stage in mitosis, it is before. Okay, this takes place before mitosis. Prophase is the first step in mitosis. Okay, so before mitosis takes place, DNA replication needs to take place, and you will learn this in grade 12. The DNA is copied, okay, and the chromosomes double up. So they were just one before, they replicated themselves to become doubled up, okay? So during prophase, what happens is before this takes place, your chromosome or your nucleus will look something like this. The chromosomes will be entangled inside and not distinct. You will not be able to see the chromosomes. So during prophase, the chromosomes start to shorten and they become visible. You'll start to see them. Those small little X's will start to become visible. Remember, cells have 46 chromosomes altogether, but for the sake of making this more understandable, we're just going to look at 2. But in all the cells in your body, you've got 23 X's. You've got 23 chromosomes, 23 pairs of chromosomes, which make 46 chromosomes altogether. But for the sake of making this simplified, we only use 2. Okay, so what happens during metaphase? During metaphase, think of M middle. Okay, they line up in the middle. The chromosomes line up in the middle. We call that middle the equator. Okay, cells line up in the middle or in the equator. Okay, during anaphase, think of A for away. Okay, during anaphase, the chromatids are pulled apart or away from each other. They are pulled apart. So think of A for away or A for apart. So during anaphase, the chromatids are pulled apart from one another. The one will go onto this side of the pole and the one will go into the other part of the pole. Okay, this is known as the centromere. This is known as the centrosome, and these are spindle fibers. Okay, which are formed during prophase actually. So you can add it there. During prophase, the spindle fibers and the centrosomes, and the centrosomes become visible. Okay. During telophase, the cell pinches in the middle. So what we call a cleavage will form in the middle. Okay. And then two identical daughter cells will form. You see, this daughter are identical to that parent over there. They're identical to the original cell. So what we did was we just produced two out of one. So we doubled this up to form two cells that look exactly the same. Okay, so I'm gonna quickly I know we are out of time, but I'm gonna quickly repeat this. Okay, let me erase everything together. Okay, to remember this, we remember it as PMET. Okay, these are the 4 phases of mitosis. In prophase 1, chromosomes they shorten and they become visible. Okay, and then spindle fiber starts to become visible, as well as the centrosomes, they start to become visible. During metaphase, the chromosomes line up in the equator. During anaphase, the sister chromatids are pulled away from each other or pulled apart. During telophase, a cleavage is formed and two identical daughter cells are formed. Cytokinesis, the cleavage will be divided and each nucleus will be pulled apart from one another. Okay, so that's the stage of mitosis. You can, by looking at this is what stage do you think this is? What phase of meiosis do you think this is? Where the sister chromatids are pulled from one another, they are pulled apart or pulled away from one another. What stage of mitosis is this? This is anaphase. Remember PMET, A for apart. When the sister chromatids are pulled are pulled apart from each other, we call this phase there the anaphase. Okay, so you guys must please look at the typical questions. Please go and study and try and attempt the questions from the slides, as well as do corrections from your past papers. Good luck, and I'll see you next time. Remember to stay safe. Bye.